TG-1701 is a Burton tyrosine kinase inhibitor, and this was a phase 1 trial exploring the safety and activity of this agent both as monotherapy and in combination with the PI3 kinase inhibitor Umbralisib and the anti-CD20 monoclonal antibody Ublituximab. Patients were included who had relapsed and refractory B-cell malignancies, including chronic lymphocytic leukemia. We did a dose escalation design in monotherapy with a parallel dose escalation for the combination cohort in which patients received all 3 agents. We also did dose expansion cohorts at 200 milligrams at monotherapy in specific histologic subtypes, including CLL, Waldernström macroglobulinemia, and Mantle cell lymphoma. In terms of the characteristics of the trial, patients, the median age was around 70. Most patients had between 1 and 2 prior lines of treatment, with the mantle cell lymphoma patients being a bit more heavily pretreated. And the majority had been exposed to anti-CD20 monoclonal antibodies previously. In the disease-specific cohorts, we also treated some patients who were not previously treated, including 1/4 of patients with CLL and 40% of the patients with Waldernström macroglobulinemia. The treatment was very well tolerated, and the safety profile of the agent in monotherapy was quite favourable. Most adverse events were either grade 1 or grade 2, with very few grade 3 plus adverse events and no grade 4 adverse events at the time of data cutoff. The most common ones were those you would expect to see in patients treated with BTK inhibitors, including bruising and fatigue. Of special note, we did not see high rates of grade 3 bleeding or atrial fibrillation. In the combination arm, there was a slight increase in side effects with more frequent diarrhea, as you would expect from patients treated with a PI3 kinase inhibitor, and a little bit of nausea as well, with a quarter of patients having elevations in their ALT, although it's important to note that a number of these patients were able to continue dosing when the dose of Umbralisib was reduced. In terms of activity, for patients treated with TG-1701 as a single agent, the overall response rate was 52%, and responses were seen among the range of histologic subtypes included in the study, with the exception of diffuse large B-cell lymphoma, as you would expect for this mechanism of action of this drug. And the median follow-up for patients treated in the single-agent dose escalation arm was 12 months. For patients treated in the disease-specific cohorts, the response rates were 95% for patients with CLL, 50% for patients with mantle cell lymphoma, and 95% for patients with Waldernström macroglobulinemia. In the triplet combination dose escalation arm, the objective response rate was 79%, including 22% complete responses. We saw complete responses in patients with follicular and marginal zone lymphoma, and all patients had some tumor reduction. All patients on the triplet arm remain on treatment at the moment, with a median follow-up of 12 months. The longest patient on study has been treated for just over 2 years, and the median follow-up overall is 14.3 months. So to summarise, TG-1701 has an encouraging safety profile with activity that appears similar to other BTK inhibitors. When combined in a triplet combination with Ublituximab and Umbralisib, we saw an increased activity, including complete responses in patients with indolent lymphoma subtypes. This study continues enrolment, and future registration trials are being planned.